today's, you're gonna also find a little bit more love for biotechnology and by reproduction. That's what a lot of animal biotechnologists focus about, is about reproduction. So if you have any questions and you want to ask them during the presentation, just like you can open your microphone and ask their questions, or you can put that into the chat. And I think Chloe will be the one dealing with the chat. So please, like if you have any questions, just go for it. And otherwise, we talk about any of your questions at the end. So as Chloe already mentioned, my name is Karina Gutierrez. I'm an assistant professor in the Department of Animal Science, and I'm in the area of applied embryology and genomics. So here, just in this picture at the entrance, you can see here, it's a picture of a oocyte. So nice to meet you to everyone that they don't never seen an oocyte before. This is an oocyte. And in here inside this injection pipette, we have a little sperm. So just out of curiosity, the oocyte is the largest cell in the body. So it's just one cell and it's big as that. And the sperm is also one individual cell and it's a really tiny one. So we can see how the oocyte, it's a big cell. And the oocyte is the female gamete, for those who don't remember from high school or still didn't get to that point. So is the female gamete in the body, and the sperm is the male gamete in the body. And here are some of the pigs that we generated during my PhD here at McGill, and a little picture of a marmoset, which is a non-human primate that we are also studying right now in the laboratory as an animal model. So a little outline of this presentation. I'm going to talk to you guys about my background. So what made me choose what I do and the pathway that I had to go through. It doesn't mean it was the right or the wrong one. It's the one that worked for me. And I hope it can inspire you if that's also your goal, to study biotechnology or science in general. So I'm going to talk a little bit about biotechnology, some of the animal models we are using currently in the lab and why it's important to have those animal models. And a little bit, I'm just going to briefly touch some of the research we have been doing and that we did in the past years. So as a background, you might notice by my accent and my English that English is not my native language. So I'm from Brazil, and Brazil is a vast and big country. And it's actually the only country in the South America that speaks Portuguese and not Spanish. So contrary to many people think that we speak Spanish, no, we speak Portuguese. So we are known by our huge biodiversity and diversity. My state is in the south of Brazil, and it's the state where we have the Iguaçu Falls and we have huge productions of coffee beans. So in the 60s, the coffee beans were known, like in my region, as the green gold, because that's what brought lots of money for the region. So my hometown is a small town that's called Apucarana. It's the indigenous name, which means Wuthering Heights. And we are known because we have a huge population of those cute animals, the capybaras. I know they are very in and hype like in the moment, and my city has lots of them. So here we can see a park for kids that's being just used by the capybaras to have and spend some good time in there. We even have some foods that are made in capybara shape. So as growing up as a kid, I had a very unusual dream as a profession. So I want to be a glass blower. So many of you might know the profession or not, because maybe you're not into that, but there is a series on Netflix that's called Blow Away. And that's when the profession became really known, like in the past few years. But that was my dream. I was obsessed with those little animal-shaped crystal glass formats. So that was my dream, to become a professional glass blower. And then when I was around 10 years old, I was watching TV once in the morning, really early in the morning, around 6 a.m. And I saw it was a program, a TV show that was about agriculture and business, and very not like a kids program, but my dad loved to watch it. So I watched it along. And they were talking about a farm that was improving all their system of reproduction, and they were expanding, and they were getting lots of money like in their business because they were investing a lot of technology. And that technology was embryology. So they were creating in vitro embryos of their best cows and selling those embryos. So here we can see inside that plate, here's a oocyte, all those things moving around our little sperm. So that is in vitro fertilization. And after that, we're going to end up with a structure that is a blastocyst. So it's an embryo. And with those embryos, they could have the production of those blastocysts and vitrify them, so freeze them at very low temperatures and ship them all around the world. And then farmers could buy those really good produced embryos and improve their farms as well with like the products that those cows would be producing. So the 10-year-old me thought like, I want to be an embryo scientist, because for me it was amazing that you could make a little cow inside the laboratory in a petri dish. And what I think that those two professions have in common is like lots of creativity, persistence, patience, and passion. So that's what the 10-year-old me thought that would be the best profession for me. And that's what I kept and tried to do. So when it was time to enter in my bachelor degree, so entering the university, I decided to do a bachelor's degree in biology, in biological science. So I did it from 2007 until 2010 when I got my degree. And during my first class, one of my professors told us that the university is a place where you also attend to class. And that stick with me because I thought I was there to like go to my class, make some friends, have a good time, but in the end get my degree. And from that first lecture, I learned that no, I could do more inside the university. So that's when I decided I wanted to do a training in some labs and try different things until I find something I really liked. So the first thing I did is I joined a laboratory in the Faculty of Medicine, in the Department of Hematology and Oncology, and it was a laboratory for cytogenetics and molecular biology. So that was my mentor, Dr. Virginia Korzer. And what I was doing was karyotyping to study and to see patterns and create diagnoses for chronic myeloid leukemia and to find chromosome abnormalities in those patients. So it was inside the pediatric unit. So we had all like young children and infants as patients. So I spent 1 year in that laboratory doing lots of karyotypes. And I loved molecular biology. I learned a lot in that lab, but my inner kid wanted me to become an embryo scientist. So on the end of that year, I decided to join a lab inside the same university, but now instead of being the Faculty of Medicine, inside the Faculty of Veterinary Medicine. And that's when I did 3 years pretty much of internship inside that lab. So those were my mentors, Dr. Padilla, Dr. Gonçalves, and Dr. Oliveira. And that's when I started to study reproductive biotechnologies and molecular biology. So the name of the lab is BioRep, which is biology of reproduction. And I really was like, I was really taken by the possibility of creating embryos in vitro and making my dreams as a kid become a reality. That was also the place where I had my first publication, scientific publication in a journal, and I learned a lot about teamwork and friendship. And the friends I made during that time, I still carry them along and they still take a special place in my heart. So after my bachelor's degree, I decided to do my master's in animal science, again in the same laboratory in the Faculty of Medicine and with the same mentors, Dr. Oliveira and Dr. Gonçalves. And that's where I studied molecular biology and reproductive biotechnology using the mouse as a model. So during my undergrad, I used the cow as a model. Then in my master, I used the mice as a model for the testicular degeneration and spermatogenesis and stem cells, like reproductive stem cells. And that was my publication for my master's. And concomitant to my master's, I decided to start a degree in education because I loved teaching and I loved talking to students and creating course for students. So I joined a degree in education in the Faculty of Education. And Dr. Araujo was my supervisor during that time. And it was a very multidisciplinary approach For everyone that had a bachelor degree in science and wanted to explore even further education, they would join this program. They would create courses to improve the education of students that are in professional course. So the thing we did was create hands-on training and activities for students for the FMT programs, so farm management and technology programs. And here's me with all my friends that were from the veterinary school. And those were our students from the FMT program, where we developed hands-on training for those students. So after I finished my program in 2014 in education, I decided I want to pursue something more, and I still wanted to work with lots of embryos. So that's when I decided, like, why not go abroad and try something new in a different language, all like very far away from home, and like, let's see how it goes. And then in 2014, January of 2014, I came to Canada to do my PhD in animal science at McGill University under the supervision of Dr. Bourdignon and Dr. Agelan. And I studied refinements of CRISPR-Cas9 gene editing and its applications to study porcine early embryo development. So that is where I had my first experience studying gene editing using the CRISPR system, where I had experience with lots of biology, like molecular biology techniques, such as mRNA synthesis, gene expression, and I could apply a lot of biotechnology and embryo development techniques, such as ICSI, IVF, PA, and SNT. Don't worry about all these acronyms, you're gonna get to them later on. And that's when I also learned lots of embryo microinjection and embryo production. So here we can see some clones that we did of mini pigs. So this little pig is a clone. Here were the first piglets that were generated with CRISPR-Cas9 here in Canada during my PhD. Here's just the techniques of micromanipulation. We also had some experiments involving heifers. And here's the amazing team that follow me, like all my colleagues and lab mates that helped me a lot during my experiments in my PhD. So after my PhD, I started my postdoctoral training, again, under the supervision of Dr. Bourdignon here at McGill University. And I stayed in as a postdoctoral training from 2019 until 2023. And I studied again, I continued developing and improving the gene editing mechanism and techniques using CRISPR-Cas9 in embryos. So I was also studying embryo development, biotechnology, and in my PhD, the focus of the gene modifications we were creating was to create biomedical models, so animals that could be used to study diseases and human conditions. And then later on my postdoctoral training, what we were doing was studying gene modifications we could create to improve animal production and also to improve animal welfare. We also start to develop lots of LOPUs. So this is a procedure where we can recover oocytes from a live female. So instead of getting ovaries as samples, we could collect from the females itself. And I also started working with the non-human primates, the marmosets, to study embryo development and try to do some gene editing techniques. During that time, I was also, and I'm still is a reviewer for the Pedagogical Merit Review for the Animal Compliance Office of McGill University, together with the Canadian Council of Animal Care, regarding the ethics and use of animal in research and teaching labs. So what I showed to you is just like my history, and I want to point some real life advice. So the first one is, you notice that I spoke about a lot of my mentors and people that guide me throughout my journey. And probably those people mean nothing to you because you don't know them, you have no idea about their research, but to me, they mean the world. So that's what I want to say is like, choose your mentors and choose them well. So that does not apply only to supervisors, it applies to friends, to students that are 1 year ahead of you, that already know about like how the journey in university goes. So it's really important to choose people you want to be surrounded by, and you wanted those people to motivate you. So it's really important. Another piece of advice is choose a career that aligns with your interests. And that's because we will change during the time. So the 10-year-old me wanted to be an embryo scientist, but had no idea how to get there. And interest changed, but I kept it true to myself. That's what I wanted to do, and that's what I'm doing now. So choose something that you love But expect that maybe your pathway to there, it's not going to be as you expect. But if you know what you want and you know what your interests are, you're going to get there. And another piece of advice, again, not only applied to profession, but to every aspect of your life, is don't compare yourself to others, because you never know. You might be comparing yourself to someone that's really good to someone that's not as good, or that they like, you don't know the whole history of that person. So compare yourself to your older version and try to be better and improve every day. So now talking about biotechnology, that's what we came here for, right? So here I want to introduce you to Cowpea, this amazing creature that I made in Excel, very poorly designed. So what I want to talk about is about GMOs, so the genetically modified organisms. So sometimes they're very scary, and people might think they will be this Frankenstein creature that you get a cow, you get a pig, you put them together, and that's what you have, like a GMO. So for general population, not for you because you're all very educated. So I know that that's not what you think. But for someone that sees that image as a cow and a pig, and that's considered a GMO, they're going to think many things, horrendous things about biotechnology and about genetic modified organisms. What I want to point out are three things. One, the person that did this GMO here, this construction, has no idea of what they're doing. That was me, but I just want to point out that that's not the case. Like if it comes up to something like that that was created by GMO, that person that made it has no idea of what they are doing. The second thing it tells me is that biotechnology is not necessary, because what is the point of having this animal releasing the nature? Like, there's no point into it. And the third thing it tells me is that biotechnology is unnatural. And I want to say to you and affirm to you that all those three concepts are completely wrong about biotechnology. Biotechnology should be done by a person that knows what they are doing, should be ethically done, should be done correctly and in a safe way. There are regulations for it, and we should follow them. Biotechnology is extremely necessary. Without biotechnology, we don't have advance in our lives, in our daily routine. So biotechnology is extremely necessary. And here it says that biotechnology seems unnatural. And in reality, biotechnology is an evolution of the things we see in the day-to-day activities, and we just made them more consistent, and we make them happening faster. So it involves from natural mechanisms. So it's a completely natural process. So biotechnology, in my point of view, should be used as an elegant solution for our problems, like our daily problems. As we know, the population is growing worldwide, so there's a need to feed this population. So if people are living longer and it's increasing the population, we need to have like food, we need to have supplies to provide for those people. So biotechnology will enter here in terms of production to produce more food and produce it in a more sustainable way. If those people are also aging, we need to have treatments, broader treatments for more people that should be available. If people are aging, we also see, and it's being noted like in the years, reproduction problems. People are waiting longer to have kids. That can lead to reproduction problems. And agriculture, because we also need to feed those people. So biotechnology can enter in a way to help and solve all these aging problems that are being caused by the growing population, reproduction problems, and also it's improving agriculture to produce more food in a more sustainable way. So talking mostly about reproductive biotechnologies, which is what I do, like I research and what I really love, I want to present to those that never saw. So here's an oocyte, the female gamete. Here's the sperm, so the spermatozoa, the male gamete. If we put both together, oocyte and sperm, we have an embryo, a blastocyst, which later on can be a human being. So a baby can be, when you implant it, can be an animal. So a cow, a dog, a sheep, like they all pass through these first steps of life. So an oocyte, sperm, and embryo. What I want to point out here is that In the laboratory, we can produce those embryos without the need of both gametes. So in the laboratory, to be able to study reproduction, that's not really the case every time. You don't need an oocyte sperm to have an embryo. You can have an embryo with only the oocyte. But again, that's only in the laboratory settings. And another thing I want to point out is curiosity. So that's just like a fun fact. Here we have multiple pictures like microscopy from sperms from different species. So just give a quick look just to see the morphology of all those sperm, and I'm gonna point out what they are. So here we can see sperm from mouse, sperm from cat, sperm from frog, sperm from turtle, and sperm from bird. So they're all different and they adapt according to the necessities and evolution of those species. So that was just out of curiosity because that still amazes me to see how biology is so perfect. So talking about reproductive biotechnology, I already showed you the oocyte, the sperm, and the embryo. So the center of the word in reproductive biotechnology is to have an embryo and a healthy embryo. So an embryo that is able to produce more embryos, to have a system where we can produce more embryos and produce more animals out of it. If you can produce more animals, or if you can reproduce the correct animals, the animals that have the characteristics we want, the ones that produce better, or that are more calmer, or that are good reproductors. So then we can choose those animals to reproduce them, produce more embryos from them, and in that we're going to produce the products we need more efficiently. So let's consider milk. If we know the cows that produce lots of milk and the males that carry the characteristics, genetically carry the characteristics to produce more milk in their daughters, so now we can combine those two gametes, the oocytes from those good females and the sperm from those good males, produce more embryos from them. And then when we have those animals being born, we can expect more production. So more food production, if that was one of the problems we discussed later on about having more food to feed all those people in the world. And with that, we can make it in a more sustainable way because now we don't need all those animals. We can select only the good ones, the ones that carry the characteristics we want. But with reproductive biotechnologies, we can also consider another side of the biotechnology that is not only focused on production, but it's focusing on animal models. So if we know how to make it effectively to produce those healthy embryos, we can produce those animals with genetic modifications that can make them, for example, resistant to disease, to viral infections, to bacteria. We can make them resistant and not developing, not being infected by those diseases. We can also make those animals produce more of what the product we want. For example, we want cows that are going to produce more milk. We can do it. We can turn those animals into biopharmaceuticals. So what we can do is we can make those animals produce and synthesize medicine, drugs inside their systems. So for example, in cows that are going to produce insulin, and then we can recover this milk and separate, isolate insulin from the milk of those cows. So it's also possible to use them as reactors to produce pharmaceutical compounds. And we can also make them as animal models to study disease, so to mimic human conditions, and then we can study new drugs, new therapies, new surgeries, new methods to treat those conditions, or even to just understand, like, are those genes really related to the cause of those diseases in humans? So if you can recreate an animal that has the mutations in those genes, we can confirm if this gene is related or not to that condition, and we can create later on gene therapy for those conditions. So anything that is genetic could be reproduced in those animals. So remember when I mentioned that in my PhD, I studied different methods to produce embryos in vitro. And here's where I'm going to just quickly mention them to you and all their acronyms. So in order for us to have an animal model, we need to know how to produce embryos from them. So here's in the first example is IVF, which stands for in vitro fertilization. So in here, we're going to have an oocyte and a sperm, so multiple sperm that are going to be placed together inside the petri dish, and the sperm should be able to fertilize the oocytes, having them a blastocyst, an embryo in the end. We can also do ICSI, which stands for intracytoplasmic sperm injection. In that case, for example, let's suppose that the semen of that animal, that person has a problem and doesn't move forward, so it cannot swing until the oocyte. So what we can do is, with an injection, we are going to insert one spermatozoa inside each one of those oocytes, those eggs. And then we are going to do the fertilization process for the sperm. So in here, like we need micromanipulation tools, and then we can produce embryos in this way as well. We can also produce embryos via somatic cell nuclear transfer, so SCNT, which is broadly known as cloning. So I don't know how many of you are familiar with Dolly, the first sheep that was cloned using somatic cells. So what we do here is we get one of those oocytes. We are going to remove the genetic material, all the DNA from this oocyte, and you're going to insert a cell from this other individual inside, and we are going to reprogram this oocyte to become a blastocyst again. And now the genetic material is the same as the animal that donated the cell line. So in that case, when they did Dolly, they had a sheep, they produced a new sheep that had the same genetic material as the first one. So that was the most common example of cloning, but that's what we can also do in the lab to study embryo reprogramming. And another method for the production of embryos in the laboratory, and that one is really broadly used to be able to study culture conditions or to study things we can add into the media to improve embryo development, is PA, which stands for parthenogenetic activation. So in this case, we do not need the sperm. So in those two cases, for cloning and for parthenoids, we don't need the sperm because in here we are going to use the genetic material of a somatic cell, like a skin cell from an animal, and in here we only need the oocyte. When we create parthenogenetic activated embryos, what we need is chemical or like electric pulses to activate the zoocyte and tell the zoocyte, go further, you need to keep developing. And then we can produce an embryo. The thing is important to point out is that this embryo generated by parthenogenetic activation will not generate a live animal if there's a transfer of those embryos later on to a surrogated female. It's not going to become like a sheep, a cow, a pig, like it cannot develop to term, but it's really important to study the first days of embryo development until the blastocyst stage. So it's another method for the in vitro embryo production. So in our laboratory here at McGill, we use all those four methods to produce embryos to study different conditions. So here's just an example of IVF, so in vitro fertilization. So here we see the oocytes are those big round cells, and all those little dots moving around are sperm. And we can see what we call the happy dance. So when the sperm attached to the oocytes, they move so frenetically that they are able to move this oocyte around. So this is an example of IVF. So we have multiple oocytes and a lot of spermatozoa inside that petri dish to do the fertilization. And here is an example of ICSI. So here we have the oocyte, and here we have an injection pipette. Inside here is the sperm. And now we're going to mechanically insert this sperm inside this oocyte to do the fertilization. So we are passing the zona pellucida, which is this barrier in the oocyte. Now the sperm comes right here in the edge of the needle, like the injection needle. And now we're going to deposit this sperm inside. So you can see it there. The needle, like the injection, will come out and the sperm will stay inside that oocyte. So we do a mechanical fertilization. So we insert the sperm inside that oocyte. So in the lab, we currently use multiple animal models depending on the project. So most of our projects, they are using the pig as a model. We also have some projects using the cow or heifers, so younger animals as the model. We also have some projects involving the use of non-human primates, and our recent addition was the use of horse. So our goal in the lab and the way, like why do we use those animal models, is to improve fertility. So we want to study the conditions in the embryos, in the animals, to be able to improve the fertility and help, like in agricultural, for example, needs. understand embryo development, accelerate genetic gain. So for example, if you have animals that produce more, that are more valuable, we can reproduce them in vitro to maximize the number of embryos produced from those animals to be able to accelerate the genetic gain in that population. We can also generate animal models for either biomedical and agricultural purpose. So those are the animal models, and that's why we use them. So focusing on the health of those animals and in the health of those embryos that are generated. So in the end, what is necessary? So if you're talking about animal models and how to generate animal models, either for agriculture, biomedical process. So we need two main pillars, like to be able to produce those animals. We need to know the reproduction, and we need to have access to the gametes of those cells. So oocytes, here's the ovary. So we're going to have in the female. So we are going to need to have oocytes that are going to come from those ovaries and sperm from those males to produce blastocysts or have access to those blastocysts and those embryos. And you're also going to need to create gene editing. So the standard process now is via the use of CRISPR-Cas9, but could be zinc finger talents, could be homologous recombination, can be any process to create gene modification. And by gene modification, I mean we're going to either delete a gene, we're going to add a gene, we're going to modify a gene, we're going to change specific base pairs inside that sequence of DNA. So we need to combine those two things. How we do it, there are two main ways to do it. One of them is via somatic cell nuclear transfer, so via cloning. And what we do is we harvest like a biopsy of an animal. So for example, a skin biopsy, and we're going to modify those cells. So once we modify the genes we have interest, the target genes, we're going to select only the cells that have the modification we desire. Then you're going to do the cloning procedure, the somatic cell nuclear transfer. So we're going to remove this DNA from the oocyte, and we're going to insert that cell in that oocyte. This embryo is going to be activated, and it's going to be cultured in vitro until it's transferred to a surrogated female, and that animal is going to have the same modifications we created in those cells. Another way to create the gene-edited animals is throughout the microinjection. So for that, you're going to produce those animals by IVF, ICSI, if we want to produce an animal in the end, or if we just want to study until the blastocyst, can also be via parthenogenetic activation. And then you're going to perform the microinjection of those embryos with the compounds to create the gene modifications. For example, in the case of CRISPR, with the Cas plus the guides plus everything, all the tools you need to create those modifications. We're going to culture those embryos inside the Petri dish, so cleavage until they go to blastocyst formation. Then we can analyze them, or we can vitrify them, or we can transfer them to surrogate females to have the animals we want. So that's the process to create those gene-edited animals. So talking about the projects that we had in the past in the lab. So we had some projects involving buffalo where we want to improve the reproduction and accelerate the generation, like decrease the generation time between the females. So we're using lots of biotechnology reproduction tools. So doing IVF, so in vitro fertilization in the lab. We also had some projects where we want to create biomedical models in pigs using gene editing. In this case, was to create hypercholesterolemia in those animals to be able to study the condition. We're going to talk a little bit about this project soon, so I'm going to leave it out for a second. We also have some projects to study neurodegenerative and neural conditions into non-human primates. And we also have many projects involving the use and the understanding of the mechanisms, the metabolisms to decrease the stress level in culture of those embryos to improve fertility in animals. So from all those projects, I pick one to go really quickly into details because our time is almost there. So for that specific project, we used to use gene editing to reduce Bortent. So Bortent is known as the smell, strong smell and taste that comes from the products of intact males. So it's in pigs, since it's born. So because of that, since like if you do not castrate those animals at the beginning of their lives, if they grow and start puberty, they're gonna become like, when you have those meat products from those animals, they're gonna have a really strong smell and taste. And the products are not gonna be able to be sold because people are not gonna buy those products specifically. So to avoid that, a waste of products and a waste of that animal, what is done generally is that those males, the pig males, they are castrated very few days after birth. In this way, they don't produce those hormones that are gonna become negative, that will make a negative impact in their products. So what we did in this project is we created specific point mutations in 2 genes to avoid, to make sure, not to make sure, but to try to confirm if those animals that were gene added for those specific genes, they would not have, or at least have a lower levels of Bortent. And with that, we would be able to avoid piglet castration at birth. So with that, our goal was to improve welfare, so there would be no need for castration, and also to improve production, since non-castrated males, they can deposit less fat in their muscles, so they produce a healthier meat. And they also have a very good conversion rate, feed conversion rate, so it would also reduce the pollution that is created by those animals during their growth stage. So what we did is we produced embryos by IVF. We performed microinjection of the CRISPR compounds on them. We let them develop, and then later on, we can use them for transfer to see if we can confirm or not those modifications. So one thing we wanted to test was methods to improve the efficiency of the homologous recombination, the gene editing into those embryos. One thing we did is by microinjecting together with the CRISPR compounds was microinjecting RAD51, which is an enzyme that is important for DNA repair. So first thing we want to see is that by doing this microinjection, we will not affect the embryos per se. And we saw that injecting this RAD51 or not, there was no effect on the beginning of development. So cleavage is the first division that this embryo makes. So it's an important, a critical step to access the healthy of those embryos. So we saw that there was no difference in there. And also, although it was slightly high, there was no difference in blastocyst formation, so in embryo formation, but it was even better. And here we can see some embryos that were generated. And when they have the point mutation, instead of being a GTC, they are GGA. And the other mutation, instead of being a TGG, they are a TAT. So we can see that in those embryos, we have some DNA sequence that correspond to the modification one. But we were still like not satisfied with the efficiency of the gene editing. So what we did is we tested another compound which was tested only in cells. So this HDR enhancer was tested only in somatic cells in culture, and we wanted to see if in the embryos it would improve the embryo formation and also the gene editing efficiency. What we saw is that we observed a higher chance of cleavage happening in those embryos, but it was not different, and blastocyst was also not different. But those blastocysts, they look better, so they had more cell number in them. So that was a good indication that those embryos are healthier and happier because they are developing more than the control embryos. And when we wanted to test the efficiency of gene editing, we could see, so here we have like HDR only, and here HDR plus indels. So indels stand for insertions and deletions that can happen in the genes when we create those modifications, and they are random. And what we saw is that when we used in the embryo culture this HDR enhancer, we had a higher number of embryos that had those modifications we wanted, and one embryo was 100% modified in the way we wanted. So here we can see this embryo that is 100% homozygous for the modification we wanted to create. And here's an example of embryos that had the modification we wanted, but also had some small insertions and deletions going on in those genes. So I know it was very rushed, like on this experiment, so I'm more than happy to talk about them if anyone has other questions, but it was just to show how can we apply science inside like the biotechnology of reproduction. and in vitro embryo production. So how can we use science to improve that and to get to our goal? So we are still waiting for confirmation in pregnancies and animals, like to see if they're going to have less abortant or not. So we are still waiting on that. But that was just to show what we can do to study and how we use the embryos to study that. And with that, I want to say thank you. So, and I want to point another very important phrase that sometimes the solution for big problems, they're really under the microscope. So something really small can have a huge impact in our lives. And with that, I want to say thank you all for being here, and I'm more than happy to answer any of your questions.
Thank you so much for the presentation.
Thank you.
We really like the study on the pig and the hormones. I personally think it's super interesting. Does anyone have any questions relating to the presentation or the study in general? Feel free to.
Like if there was any terminology that you weren't able to understand, you can also ask a question about that. Like we can clarify it for you, or any comments as well. It's appreciated. You
Yes.
can also unmute yourself. It doesn't have to be in the chat.
Hi, my name is Chris. I just had a small question for you. I was just wondering if you found that a lot of people who are doing research are using AI models or the use of artificial intelligence in doing their research.
There are. So that is something very new into the embryology field. So thank you for the question, Kristen. So in the reproduction field, it started not a long time ago. So there are some people using it, like artificial intelligence, to either choose the best or more suitable embryos to become really embryos. There are also lots of help of artificial intelligence to help to design the experiments, although it should always be double checked. But there are also people using like in gene editing also to choose the best methods, the best time points, like to do the genome modification. So it's increasing, but I believe it's like that for all the areas. In embryology itself, it's a little bit more resistant, but it's starting now. There are grants being available now for really the use of artificial intelligence in reproduction as well. So it's a huge and increasing field, but it's still very new in the biotechnology, like reproduction.
Okay. Well, I have a question regarding your research. So I was trying to understand like the results for the graph. I think it was for slide 23.
Mhm.
Um, it's Is it that like every line is like
I
a
can
germ
put it
line?
back on just in case so everyone can know can you see my screen? Let's see.
Yeah, it's still
Yeah,
visible.
okay. The 23. This one?
Yeah, that was for the RAD 51.
Mm-hmm.
So is it that every square, is it like an embryo line?
Yeah, so this one it's a next generation sequencing, so it's called like
Mhm.
MISEQ, and each line is a strand of DNA inside that embryo.
Oh, okay.
So we can sequence all the strands. So if you think of that, the DNA is double strand break, and you have in an embryo, let's suppose like around 100 cells. So depending on the stage, between 60 to 100 cells. So it means we can have like up to 200 readings for the same embryo. So each line tells what's going on in that specific DNA, that combination of DNA. So here the
Hmm.
list goes on. It's just like a screenshot of what you can see. But you can see, for example, for some embryos, it's just like a few of them that have the modification. In here, we have more that have the modification we want. So we
Hmm.
can see like how it happens. So like all those lines, for example, they have the modification we want. That's what we want.
Oh,
So you can see like exactly where the modification is going on. And in this other one, for example, we can see that like all the reads for that embryo were exactly what we wanted. So in this way, you can really like be able to observe what's going on and what is really happening inside each one of those embryos.
hmm. Okay, that's interesting. Thank you.
No problem.
And
If you have any questions later on, if you want, like you can just email me, like feel free to get in touch.
and
Yeah.
mm,
I just wanted to say, for the contact, for example, for the email, is it an email that